Hello guys, welcome back to my channel. Today I am going to explain about viral transduction. But before that, please don't forget to like, comment, share and subscribe to my channel. Okay, so let's get started. So first of all, I am going to give you a brief introduction about what is transduction. So transduction is the process by which DNA is transferred from one bacterium to another by the help of a virus, that is also known as bacteriophage. This is a process whereby foreign DNA is introduced into another cell via a viral vector. This is basically a common tool used by molecular biologists nowadays to stably introduce a foreign gene into a host cell's genome. When bacteriophages infect a bacterial cell, their normal mode of reproduction is to harness the replicational, transcriptional, and translational machinery of the host bacterial cell to make numerous virions or complete viral particles, including the viral DNA or RNA and the protein code. Means when virus infects a bacterial cell, the aim is to produce new and more viral particles. Schematic diagram of bacteriophage, where you can see its head, its neck and collar. The middle portion is known as sheath. Then the outer covering is known as sheath and the inner portion is known as core. And you can see end plate and the fibers. Bacteriophage basically infects a bacterial cell through its fibers. Okay, so phage composition and structure. It is composed of nucleic acid and protein. And its structure comprises of head and tail. Okay, so there are basically two cycles, the Lytic cycle and the Lysogenic cycle, which occurs when the virus infects a bacterial cell. So the Lytic cycle is basically referred to as a cycle in which the cell is completely destructed, and the lysogenic cycle refers to where cell remain dormant for thousands of generations. So if the lysogen is induced by UV light, for example, the phage genome is excised from the bacterial chromosome and initiates the Lytic cycle, which culminates in the lysis of the cell and release of the phage particles. The Lytic cycle leads to the production of phage particles, which are released by lysis of the host. Okay, so assay for Lytic phage. Lytic phages are enumerated by a plaque assay. Plaque assay refers to a clear area which results from the lysis of the bacteria or complete destruction of the bacteria. Each plaque arises from a single infectious phage. When a phage infects a bacterial cell, the infectious particle that gives rise to a plaque is called a plaque forming unit, that is shortened as PFU. So there are several examples of this, like P22 phage infects Salmonella chlorosis, lambda phage infects E. coli, T4 phage infects E. coli, and so on. So this cycle basically involves 5 steps. First is attachment, in which phage attaches to the surface of the host. Second is penetration, in which the viral DNA enters the host cell. Third is biosynthesis, as clear by the term. Phage DNA replicates and makes phage proteins. Maturation, in which new phage particles are assembled or produced. Lysis, in which the cell lyses and releases the newly made phages. Okay, so I have included some important terms which are important for you to know. So first is lytic or virulent phage. So these phages which can only multiply in bacteria and kill the cell by lysis at the end of the life cycle. And eclipse period. So you might be aware of this term period. So during the eclipse phase, no infectious phage particles can be found either inside or outside the bacterial cell. That is, the phage nucleic acid takes over the host biosynthetic machinery and phage specified mRNA and proteins are made. That means there is an orderly expression of phage directed macromolecular synthesis, just as one sees in animal virus infections. Early mRNAs code for early proteins which are needed for fast DNA synthesis and for shutting off host DNA, RNA, and protein biosynthesis. Whereas in some cases, the early proteins actually degrade the host chromosome. So after fast DNA is made, late mRNA and late proteins are made. The late proteins are the structural proteins that comprise the phage as well as the proteins needed for the lysis of the bacterial cell. I hope that in a healthy bacterial cell, early mRNA and early proteins are made, whereas when bacteriophage infects bacteria, late mRNA and late proteins will be produced. Okay, so next is intracellular accumulation phase. What is intracellular accumulation phase? So in this phase, the nucleic acid and structural proteins that have been made are assembled and infectious phage particle accumulate within the cell. Next is lysis and release. In this phase, bacteria begins to lyse due to the accumulation of the phage lysis protein, and intracellular phage are released into the medium. Okay, so this is the diagram of Lytic and Lysogenic cycle to understand that how these cycles differ. There is one basic difference in Lytic and Lysogenic cycle, that is in Lytic cycle the cell will completely be destructed, and in Lysogenic cycle the cell will become dormant for thousands of years. That is, the cell just becomes inactive. It is not completely destructed. Okay, so I hope you understood everything. If there are any doubts, feel free to ask in the comment box below. Okay, so that's it. Thanks for watching. Don't forget to like, comment, share and subscribe to my channel. Thank you.